Great. Thanks very much, Pedro. Thanks to the organizers for the invitation. This seems like a very interesting and hopefully a very impactful webinar that I wish back in time when I was writing this application I would have had. So unlike Paula, I'm quite an old guy. My journey started back in 2015. So like all of you, I moved from Ireland to Denmark to join Jesper Andersen's research group at the Biotech Research and Innovation Center at University of Copenhagen. And of course, when I joined the lab, I was assigned to a project already. It was focusing on epigenomics and specifically epigenome targeted therapies for cholangiocarcinoma. So then for around 4 months, I was working on that project and I defined a specific question I would like to write the Marie Curie about. And this also included, as Paula alluded to, the generation of some preliminary data. So then what happened was that we went into a mandatory one-day Marie Curie workshop. So this is compulsory for anyone wanting to submit from University of Copenhagen. They take it very seriously. And apparently today, this course has actually been escalated from a one-day event to a 3-day event. And essentially what they did during this was to bring us through each of the sections, what they mean, what a successful application should look like. They asked us to sit with our peers at the time and to talk about what they liked and did not like about each of these sections. And we also heard some interviews from previous Marie Curie evaluators to tell us exactly how things work and how the scoring system works. So after that, in July and August, we went into internal review, several rounds with my own personal group leader. But then we also had a mandatory internal review by a University of Copenhagen grants officer. So again, certain institutes really are taking this very seriously. And then ultimately in September 2015, I submitted the application. In early 2016, we got good news that it had been awarded. And then I actually delayed it for 1 year. So it ran from February 2017 to January 2019. And then you have 2 months at the end to submit your report. So that's overall what happened to me. But the topic I'm supposed to talk about today is impact. So what exactly is impact? We all know what this means. Practically, it means to have a strong effect on someone or something. So in Marie Curie, when we're writing the impact section, essentially what you're asking is, what will your proposal have a strong effect on? And the answer is it has effects at multiple levels, but absolutely top of the pyramid is the individual fellow. So in this particular type of application, you yourself are the center of the universe. It's almost all about you. And that's one thing that makes this grant really stand out. However, of course, we would all be working on cholangiocarcinoma for this audience. So therefore, we would hope that will have significant implications for healthcare, as well as the business sectors. So that's another type of impact. And then, of course, spreading the base wider, again, we would hope that it would ultimately impact wider society itself. And as Paula indicated, really the main interaction you have to emphasize in this application is the two-way interaction between the fellow and the host lab. And what you have to be able to do is to convince the evaluator that there is extreme synergy in two directions. So you are the ideal person to solve this proposal or the problem at the center of this proposal. And the host lab itself is critical and the ideal lab to help you to accomplish these aims. However, like we know in science, nothing is really done between two parties. And of course, we are working within the context of our international network. So again, there has to be synergy between the fellow, the host lab, and the international network itself. And of course, this is an excellent setting in which the various efforts by EuroCholangioNet can help you. So coming into this type of application, I did find at the time it was quite a black box. We didn't really know what was going on. Of course, the University of Copenhagen course really helped us. But in preparing this slide today, I decided actually to start from the end, which is to show you the evaluator sheet that everyone gets after you submit a Marie Curie fellowship. So each application is independently reviewed by 3 evaluators. Then the evaluators meet to assign an overall score for each of the 3 sections. Each section is initially scored out of 5, and then overall scores are assigned using adjusted weights per section. So this is impact. The impact section counts for 30%. As Pedro discussed at the start, you have a very strict 10 pages to keep to. You cannot go outside those bounds. We're all scientists or researchers, so we can talk about our topic all day, and we can certainly fill up 10 pages of excellence. But really the most remarkable thing and the biggest struggle I had was actually cutting that back. So the excellence section only counts for 50% of the scores. In theory, then it should somewhat only account for 50% of the space. In my particular case, I spent 5.5 pages on the excellence, and then I distributed the remaining 4.5 between these two sections. Now, impact is 30%, whereas implementation is 20%. So in theory, impact should be a bit longer than the implementation. However, at least from my own personal experience, I found the implementation section to take a larger amount of space. So in the end, I ended up dedicating approximately 1.5 pages to this section on impact. So going through the various different comments I got on the impact section, we think that we can tell what exactly the evaluators are being asked to judge by. So I was only the second person in my host group to get a Marie Curie fellowship. Since then, we've been very successful. And every time someone gets a fellowship, we go through the evaluator comments, and we find that the same words and phrases are repeated over and over and over again, which means they're probably what specifically they're being asked to judge on. So we think that hopefully now we've cracked the blueprint to help anyone succeed in scoring high marks here. So the first thing that they commented on was competences and skills that the fellow, in other words, me in this case, will obtain. So upon recommendation of our course at University of Copenhagen, we were told to use very specific terminology in each of these sections. So here you can see these in red. I had a little section written in each of these. So the first one was to develop new knowledge bases. So in other words, I was moving from a background in a different type of cancer to cholangiocarcinoma. So I would expand my knowledge base into cholangiocarcinoma, which is the expertise of the host lab. And on top of that, I would also expand upon my own pre-existing skill sets in epigenomics and biomarkers. So I was already continuing to develop something that I had established myself in my background. Next, they specifically liked the accrual of new technical expertise. So previously, I indicated the skills that I was coming with. Again, what was I bringing to the lab, kind of what Paula alluded to. And then I also indicated specific skills that I would be learning completely new from the host lab. So in this case, it was CRISPR-Cas9 and in vivo modeling. And lastly, moving a little bit away from the more academic setting, I also had a section on establishment of leadership skills. So I wrote that I would take specific local courses in scientific writing, as well as management and leadership in academia to try and advance my basic skill set, my interpersonal skill set. I would take over supervision of PhD students very formally. I would work closely with my PI to drive career progression from the early stage postdoc that I was at time of application to more a senior postdoc, and ultimately to become a principal investigator myself. And then lastly, I also indicated that I will be participating in a lot of outreach and dissemination, but we'll talk more about that shortly. They also commented on new collaborations, environments, and mentoring. So in terms of the new environments, I had moved, as had anyone applying for a Marie Curie. I was entering into the Anderson Group, which has a very multidisciplinary expertise. So it spans clinicians to computational biologists, in vitro and in vivo experimental biologists. But not only that, I was moving into a host institution that has a wide variety of different research topics, as well as multiple state-of-the-art core facilities. And again, this is overlapping with the section you'll hear about next, which is implementation. So the next positive thing they commented on was new mentoring. And in particular, they seemed to like the international research background of my principal investigator. So again, they do place a lot of emphasis on the specific mentor you'll have during this time. They also like the interdisciplinary connections of the PI, including his role in establishing the European Network for the Study of Cholangiocarcinoma. So this was very, very early days, but already we had indicated this. And lastly, they positively commented on new collaborations. So we'd have local collaborations here in Denmark and a lot of other encounters, both through collaborations with PhD students and disseminating to work on. So next, we were also recommended by our university to include a specific section called Impact on Employability. So this is to advance my or your new career perspectives. So under this section, I commented on the potential to expand my knowledge base as well as my skill sets, increase my multidisciplinarity, as well as develop my career profile as a nationally and internationally competitive researcher in translational cholangiocarcinoma omics. So really what you want to be able to emphasize is how this very unique opportunity that you are uniquely suited to will significantly impact your professional development. And then lastly, and this is one of two things we were very surprised about, the evaluators went out of their way to comment on the fact that my supervisor had collaborations with industry. And this was impactful for two reasons. One, they were associated with the project, which was on epigenetic inhibitors, but also they linked it with a career perspective. In other words, because my supervisor had these connections with industry, then perhaps that would be an excellent segue for me to move into industry if that was the career route I would like to follow. And we found this particularly interesting that they commented on, because I think I only had one line stating this and still they caught on to that. So certainly something to bear in mind. So moving a little bit away from the individual fellow to wider impact, the next thing to comment on was the impact of the project on healthcare and business sectors. Now, for all of us applying from a cholangiocarcinoma perspective, this is quite straightforward. Obviously, it still carries a devastating prognosis, many unmet clinical needs. In my specific case, they commented specifically on the topic. So I was working on epigenetic therapies, and there's quite a lot of different targets there. There's quite a lot of different individual drugs. And when you consider those in different combinatorial treatment strategies, there's many, many permutations. So in theory, successful accomplishment of this project would impact multiple of these pharmacological approaches. Of course, the context of my application was cholangiocarcinoma, clearly a rare orphan disease with many, many unmet needs. However, it was theoretically possible that the new insights gained from this project will also impact other cancer types. And that's certainly something to bear in mind and comment on for the new applicants listening. And lastly, upon successful completion of the project and assuming that they impact both the context and the topics at hand, they would have significant ramifications for healthcare and business sectors in Europe. And that's really an important link that they're looking for. So then moving lastly into the societal impact, they commented on the dissemination plan. So they said it was very well presented and targeting two different types of audience. So the first type of audience was the general audience, and this is the outreach activities that you have to complete. So in total, we were recommended to do between 2 and 3 outreach activities. So we went for 3, though they were not of all equal effort or input, as you can see. So in the first case in Denmark, we decided to participate in a government-funded initiative, and that's when high school students can come and they can actually write a research proposal and try to carry out some of these basic experiments in the lab with you. So we did this. This was a lot of fun, and in this case, we were of course aiming at teenagers. Next up in Denmark, they have something called Annual Inspiration Day. So this is a way to inspire high school teachers to bring new ideas and new approaches to their teaching classes. In this case, we invited high school teachers from all over Denmark to come in, and we demonstrated some techniques covering CRISPR genome editing and what it can do to cancer cells. And this was also a great success. And then lastly, which is definitely the lightest of all of these, we decided to do some media engagement. So any paper or any big research finding we uncovered, we decided to work with our own PR office locally. So just on our local university website and disseminate the research findings in simple to understand language. Of course, now that we have EuroCholangioNet, that will be an excellent place to integrate some of their ongoing activities. And then lastly, as you can see here at the bottom, we did comment that at the time, the host lab was, of course, affiliated with the Cholangiocarcinoma Foundation, and that in the future, we intended to further develop patient engagement, though we didn't necessarily give any specifics. So that's the general audience. What about the scientific audience? And these are the more basic questions that we know and we're able to address. So in this section, you should indicate where exactly you will present your research. So in my case, I went for international conferences. So things like ILCA and EASL's, various different conferences. We were also told to indicate how many articles we anticipate publishing. So I said I would do 1 review and 1 original research paper. We were told to give examples of specific target journals, as well as to indicate in the Gantt charts that you will be using when exactly you would anticipate these publications to occur. And then lastly, and something that, again, they commented on that we were very, very surprised about, are you considering IPR for your findings. So we just wrote simply that when we found a new combination strategy or a new drug, we would approach the technology transfer office at the University of Copenhagen, and we'd also liaise with other groups at our institute who had successfully generated spin-outs themselves. And again, this is something the evaluators went specifically to point out. So clearly it's something they consider to be very important and remarkable. And I would encourage any of you to also include similar commentary in your own application. So with that, I will wrap up and say good luck. So some final tips are exactly what Pedro said. So I'm glad we're overlapping. One, remember you are the main focus of this call. So you always say, I will do A, I will do B. Focus on the impact of the fellowship on your role, your development, your opportunities, and your suitability. Keep the score weights in mind when deciding how much space to give to each section. Emphasize the synergy between you and your PI, your group, your institute, and your network. Make scoring your application easy for the tired non-expert evaluators. So again, using things like putting keywords in bold, underlying key locations, using keywords themselves, and also ask others to read your grant application, and preferably those who aren't specialists in cholangiocarcinoma itself. With that, I will say thanks. If you have any questions, feel free to reach out and drop me an email, and I wish you all the best of luck. Thanks.